Okay. Yay. All right. Now we're recording. Sorry, I just wanted to make sure I was recording before I did anything else. So yes, thank you guys for saying good morning. I miss you guys. I really I really miss seeing you guys. This whole this whole situation, it's at least you guys get to see me, so that's good. But I do miss seeing you guys. I hope you guys are all hanging in there. We're gonna finish the Cycads today, and you have an in-class assignment. So let's talk. We're going to talk about that a little bit. Let me, before I do that, share this first to make sure that you guys can see it. Okay. Second. Great. Can you guys see you guys can see the PowerPoint slide, right? Great. Cool. Let me go. So we covered a lot of the cycads last time. Remember the plants, that's just dinosaurs ate different aspects of the sporophyte plant. We talked about primary and secondary growth, which is my favorite topic. No, not really. But yes, we talked about the parts of the sporophyte plant that make, right, and then we talked about male and female sex organs and where they're located. All right. So yes, maybe that's a good place to recap. So I'll start around there. Before I do though, yeah, so before I do, you guys have a in-class assignment today. And so the way it's going to work is when we finish with lecture, I'm going to open the assignment on Blackboard. And it should run basically like a regular in-class assignment. Oh, I saw a, okay, cool. Let me get rid of this thing. I don't need this thing. Yeah, so it should run like pretty much basically like a regular in-class assignment. If you have any problems, contact me. I don't think you should. You just need access to Blackboard. So we'll do that toward the end of class. It'll be on the ferns. Another thing though is you and me need to kind of we need to kind of troubleshoot how exams are going to work. So initially I was going to, look, the long story short is that initially I was going to have you guys run the in-class assignment kind of similar to how the exams are going to run. But then I thought about it more and I thought to myself, it's kind of unfair I'm going to be using a different program to run exams, and you guys haven't had any experience with that program yet. So what that means is that today's in-class assignment is going to run through Blackboard, and it should be pretty straightforward. I don't think you guys should have any problems at all. If you do, contact me. But for homework, I'm going to create, and I haven't created it yet, but I'm going to create like kind of a fake exam. It'll be a silly exam, with questions like the best color is, or my favorite food is, kind of just a fake exam through the new platform that I'm going to use to take exams. And your homework assignment will be to like take that little fake exam so that you can troubleshoot whether or not the exam platform is working well on your end. So stay tuned for that. I'll post that later today. And it just will have to be something that you do in the next day or so. Shouldn't take that long. But the in-class assignment we'll do at the end of class today. So cool. All right, let's get started. There's a good chance we might finish with the Cycads early, and right, no, yeah, no. If I give you a fake exam, as it were, that is like, ooh, what's your favorite food or whatever? No, it won't be graded. The homework assignment on the exam platform won't be graded. The in-class assignment at the end of today's class will be graded, but it should be pretty easy, and yeah. Stay tuned about what platform it is. There's a few that I'm thinking about using, and I haven't decided yet which one I want to use, which is why I haven't posted the fake exam yet. There was a question about what platform is it. Okay, so last class we talked about the Cycads. So we'll recap here just a little bit. Cycads are strictly dioecious, right? There's male plants and there's female plants. And the female plants always have their megasporangium, right, in an ovule. And those ovules are located either on a crown of fertile leaves, which we talked about last time, or a strobilus, right? So last time we went through this, so we won't go through this too slowly because we already went through this last time. But this is an example of two female plants from two different species. One has a crown of fertile leaves, and one has a strobilus. Come on, here we go. Right. This is a close-up of a strobilus on a female plant. So this species has a strobilus, and the strobilus is a stem, leaves, and in this case, ovules, because it's on a female plant. And each leaf of the strobilus doesn't really look like a leaf. It looks like this weird thing, as you can see in this picture, but they are technically leaves, and each leaf bears a certain amount of ovules depending on the species. This is, on the other hand, an example of a female plant that bears her megasporophylls in a crown of fertile leaves. But similarly, you know, the leaves are still organized. They're organized in a crown, and each leaf still has a certain number of ovules attached to it. The number of ovules will depend on the species. So, as we talked about last time, the megasporophyll shape looks really different depending on what species you're talking about. So, for species who have their ovules organized in a strobilus, like the ones here in blue, the leaves are usually really, really small and short. That's because they have to fit onto a strobilus. Remember, a strobilus, you know, kind of is a way to kind of condense the structures. So if the megasporophyll leaves are on a strobilus, they tend to be really small and compact. Whereas if they're part of a fertile crown, they often tend to be more leaf-like in shape, right? Like on the left here. Let me pull this up on here so I can see where we're going. One sec. There we go. Okay. Right. And as we talked about last time, the number of ovules per leaf will depend on the species of cycad. So some cycad megasporophylls might only have 2 ovules per leaf, whereas some other species might have as many as 6 or even more than that. There we are. And then here's around where we left off last time, about talking about where the male structures are located. So unlike the megasporophylls, which are sometimes arranged in a strobilus and sometimes arranged in a crown of fertile leaves, depending on the species, the male plants always have a strobilus in cycads. Always, always, always. So this is just a reminder of what makes up a strobilus. And we left off here last time to look at the different parts. So here on the left is the example of what the male strobilus might look like. If you were to, so here's a diagram of a male strobilus. If you were to pull off a leaf of this male strobilus and look at it up close, the microsporophyll, right, the leaf of the male strobilus, it would have a bunch of microsporangia on the bottom of the leaf, and the microsporangia on the leaves look like this, depending on species, but in general, they kind of look like this. They're a lot smaller than the ovules. And there's lots and lots and lots and lots of them per leaf. All right, so this is where we left off last time. And now we need to talk about, let me make sure I have the clock here. Cycad reproduction. So we know where the sex organs are. We know what the sex organs produce, right? An egg, in terms, the ovule produces an egg. The Microsporangium produce microspores, which basically produce sperm. But now there's a little bit of a difference. So this is a recall slide, right? In cycads, the male gametophyte plant is even more reduced than we saw before. So recall that when we looked at heterosporous species for the first time, which was in Selaginella, the male gametophyte plant was what I used to call testicles in space, right? Thanks, Dr. Casper. I can never not think of it that way. But the male, the microspore was basically a structure with a single Antheridium inside, right? That's what we saw when we first learned about the heterosporous plants in Selaginella. Microspore with a single Antheridium inside. Now the male gametophyte plant is reduced even further. It's not a whole Antheridium inside of a microspore anymore. It's even smaller. So we'll talk a little bit about how or what that means. So this is another evolutionary leap. When we're going from when we see reduction in the gametophyte plant along the different divisions of the plant kingdom, every time it gets smaller, it's another evolutionary leap. So in the Microsporangia, there are Microspores Sites. We're used to that now. And the Microspores Sites are diploid and undergo meiosis and cytokinesis in order to produce a tetrad of microspores, right? That part we're used to. Those microspores are going to undergo mitosis and cytokinesis to become this highly reduced male gametophyte plant, which we now call the pollen grain. So, major evolutionary leap for the megaspore was that now in the seed plants, it's inside of an ovule. Major evolutionary leap for the microspore is that now it contains a even more reduced male gametophyte plant, which we call the pollen grain. And in cycads, that pollen grain has these parts: a prothallal cell, pollen tube cell, and a generative cell. The prothallal cell, we don't really know what that even does. We think it might be leftover or vestigial from the shape of a prothallus. We're not really sure. But there is a cell there that we call the prothallal cell, but we don't really know what its function is. The pollen tube cell, as the name suggests, grows a pollen tube, which we'll talk about. And the generative cell is what generates the sperm. So, major evolutionary leap. Megasporangium in the seed plants is now in an ovule. Microsporangium produces microspores that contain an even more reduced male gametophyte plant that we call the pollen grain. So here's a picture of a pollen grain, and this is a picture of cycad sperm, and similar to the plants we've seen so far, cycad sperm still swim. However, unlike the primitive plants we've seen before, when the cycad sperm swim, they don't swim through water. We'll talk about that a little bit more as we kind of develop this concept. Now the sperm swim down the pollen tube. And as you can see from this microscope slide, each sperm contains many, many cilia. So cilia are like these little hair-like things that help the sperm swim. And you can see on this slide of a cycad sperm, all of these little tassels on the right-hand side are the cilia. So many, many, many cilia. Yep. So there's a question, is that just one sperm? Yes. So that's a single cycad sperm, which is part of the pollen grain. So that image shows one single sperm. So let's talk a bit more about this, how this all works, since this is a new concept. Let's kind of go through this step by step in terms of the pollen grain. So here is a diagram, and we're going to go through it step by step. So first we have this microspore, which contains, right. So this microspore would have been the result. Let's go back real quick. Right. So remember, you have a microsporangium, which in cycads looks like this, right? Each of these dots on the bottom of this leaf is one microsporangium, right? Each of these dots. So this leaf has, I don't know, whatever, 20 microsporangia. Inside of the microsporangia, you have the Microsporos sites that are 2N that will undergo meiosis and cytokinesis to produce microspores. Right. So there's a question, what's the difference between a pollen grain in cycads and the Antheridium of Selaginella? They both produce sperm. Indeed, they both do produce sperm. And the difference is that the pollen grain, so the difference is, so the same thing is that they're both considered the microspore, for better or worse. The microspore in Selaginella contains an entire Antheridium organ. The microspore in cycads and the other seed plants contains much less content. In the case of cycads, it's prothallus cell, pollen tube cell, and generative cell. So a couple cells as versus an entire Antheridium organ. But let's go through this now and maybe it'll make a little bit more sense. So yes, there's microspores on the bottom of the leaves that make up the male strobilus. Inside of each microspore are Microsporos sites that are 2N that will produce microspores. So that's where we are here. This is one microspore of the tetrad of microspores that would have been made by the Microspores site. This microspore is going to undergo, so this is all happening, this part right now is all happening inside of a microsporangium. So you have the microspore. The microspore is going to undergo mitosis and cytokinesis. And the result is a pollen grain. So on the left, you have microspore. On the right, you have the finished product, which is pollen grain. The mitosis and cytokinesis that occurred allowed the microspore to go from a single-celled structure to a three-celled structure that's all contained still within the same cell wall. And so what it contains is, as I mentioned, this prothallus cell, which is at the bottom, which we still really don't have a good handle on what that even does, and we think it might be just leftover from evolutionary processes that occurred before. This pollen tube cell, which is what's going to grow the pollen tube, and then this generative cell, which is what's going to generate the 2 sperm. When the pollen grain lands on an ovule, first thing that's going to happen is this pollen tube cell is going to grow. And it's going to grow into the ovule, which we'll talk a lot more about that later. But I'm just trying to get you comfortable with the parts of the pollen grain right now. But when the pollen grain, aka microspore, right? They're kind of like, I think there's a question in the lab about what's the difference between a microspore and a pollen grain. Well, the difference is that a microspore is a single-celled structure, whereas a pollen grain is a multi-celled structure. However, the pollen grain is the result of a microspore undergoing mitosis and cytokinesis. So both of them are kind of part of the male process, but they're at two different stages. When the pollen grain lands on the ovule, pollen tube cell is going to grow down, and the generative cell is going to create two sperm, which can then swim down the pollen tube cell. And I know I showed you a picture of it on the previous page, but then this is what the cycad sperm look like. And you can see the cilia kind of radiating around the edges of it. Lots and lots of cilia. Okay. So because the Microsporangia produces ultimately the pollen grain, another word for the Microsporangia is pollen sack. So there's a question, so that long figure is pollen. Let's go back. So this structure here, ooh, I should annotate. Let me see, yes. Ashley, I should annotate. So this whole thing is a pollen grain. The pollen grain has three parts. Prothalial cell, here's the nucleus of the prothalial cell. We don't know what prothalial cell does. Generative cell, here's the nucleus of the generative cell. Generative cell is going to create two sperm cells or generate two sperm cells. And pollen tube cell, here's the nucleus of the pollen tube cell. It's going to create the pollen tube. When this whole structure lands on an ovule, the first thing that's going to happen is this pollen tube cell on the top is going to elongate and grow into a tube shape. And that tube is going to lead down to the ovule, or into the ovule rather, where the egg is. And the two sperm cells are going to therefore be able to like swim down the pollen tube cell. So pollen grain has multiple parts. One of those parts is the pollen tube. Okay. Again, if you hear a banging noise, that's my heat that I have no control over here. Okay, so because the Microsporangia ultimately release pollen grains, we call the Microsporangia also pollen sac. It's the same thing. Calling it a pollen sac at this point just kind of shows that we're in the seed plants where the egg is inside of an ovule and the sperm are vectored via the pollen grain. So when the Microsporangia open to release their products, what they open and release are pollen grains. This whole process here, right? Going from Microspore to pollen grain occurs inside of the microsporangium. And when the microsporangium or Microsporangia, if there's many of them, open, it's the pollen grain that comes out. Okay. So therefore, another word for the Microsporangia in the seed plants is pollen sac. So yeah, so this whole thing here, this whole structure here is one leaf of a male strobolus, and each of these little Pac-Man dudes on it are each a microsporangium, aka pollen sac, because now they're releasing pollen grains. Cool. All right, so what do we have left to do here? Okay, not too much. That's good. So what we're going to do now is we have learned the players, and we're going to go through alternation of generations as it pertains to the cycads. And I will say that a lot of the steps we're going to go through today also pertain to other gymnosperms, meaning ginkgo and the conifers. There's some slight adjustments when we talk about ginkgo and some slight adjustments when we talk about conifers, but the basic strategy of alternation of generations in cycads is the basic process that occurs in all of the gymnosperms, right? Okay. So let's do it. So what do we talk about first? We talk about both and then go into the female. So cycads are strictly dioecious, right? 2 houses, 2 plant individuals. Some have male sex organs, some have female sex organs. And so here you go. You got 2 cycad plants. Here's a female cycad plant, sporophyte plant, cycad, and then on the right you have a male sporophyte plant, right? Obviously they're sporophytes, so obviously they're diploid. 2 plants. Let's see, do I have it lined up? Yeah, so female on the left, male on the right. So on that female plant, let's say that this female plant bears their sex organs in a strobolus, not a crown of fertile leaf. So this species has strobolus. Each strobolus, which is 2N, has leaves or megasporophylls, which are 2N. And each of those megasporophylls bears ovules, which are also 2N. So here is a close-up of a megasporophyll, right? Here's a close-up of the leaf, this thing here. Ooh, I'm getting better. Oh, look at that. Wow. That thing there is the leaf. Is a leaf that's like, let's say that's this leaf right here. Pulled off of the strobolus. And I could change color. Boom, boom. This is the top of the leaf here, or adaxial side. This is the bottom of the leaf, or abaxial side. Remember, a handy way to remember that is ab with a B is bottom, bottom of the leaf. And on each leaf are, in this case, 2 ovules. Here's an ovule, and here's an ovule. Whoa, oh, I'm getting so much better at annotate. Glad I didn't give it up. All right, so if we were to look at one of these ovules, oops. Hey, what happened? Oh, I didn't realize. Okay, yeah, sorry. We won't look at the ovule yet. Female strobolus with leaves, each leaf bears ovules. From a different plant, a male plant, you have a male strobolus, which also is made up of leaves. However, in this case, we call them microsporophylls because they bear microsporangia, or aka pollen sacs. And just to be clear, let's use green for a leaf. As I said before, this is one leaf. Let's say it's this leaf. This thing, not getting better, spoke too soon, is also a leaf. Let's say it's this leaf here. And on this leaf, we have 2 ovules, because that leaf is on a strobolus on a female plant. And on this leaf, we have so many microsporangia, aka pollen sacs. This is a strobilus from a male plant, right? Okay. Right. Okay. So first let's talk about the ovule. So this is one ovule. And as we already have talked about, the ovule is made up of certain parts. The integument is the outside. There's a hole in the integument called the micropyle. And then inside of that is the Microsporangium or nucellus, which contains functional megaspore. So what does it say down here? Each ovule is diploid and it has a Microsporangium, which is also diploid, right? Just like sporangium is always diploid, right? Because sporangium is part of the sporophyte plant. And in there you have the Microspores site, which is here. And that Microspores site is going to undergo meiosis and cytokinesis to produce function. Oh, sorry, that's already functional megaspore. See, I'm getting carried away. I just want to draw and I'm not even looking at my thing. Yes. Microsporangium, inside the Microsporangium is a diploid megaspore site, which is going to undergo meiosis and cytokinesis to produce functional megaspore, which is going to then go on to do mitosis and cytokinesis to grow into the female gametophyte plant, which is always going to stay inside of the ovule. If we were to look inside of the microsporangium, this is just kind of a recap. Here's one microsporangium. As we just discussed, inside that microsporangium are the microspore sites. Microspore sites are going to undergo meiosis and cytokinesis to produce the microspores, which are haploid. Microspores are going to undergo mitosis and cytokinesis to create pollen grain, which we now have learned is the very highly reduced male gametophyte plant. So instead of an entire Antheridium, it's only reduced to a couple cells. Oops. All right. Right. All right. So that's Microsporangium. Remember from this slide, right, that the functional megaspore inside of the ovule is going to undergo mitosis and cytokinesis to produce female gametophyte. So here is that. This is the finished product. This is functional megaspore has gone on to grow into female gametophyte, which stays inside of the ovule. And I'm pretty sure that I have this color-coded already. Yes. So here you go. Female gametophyte is haploid, right? Develops and stays inside of the ovule. But the ovule itself is diploid because the ovule is part of the sporophyte plant. But now we have inside that female gametophyte plant who's haploid. Female gametophyte plant contains two archegonia, which are these little hangy things up there. The archegonia, because they're part of the female gametophyte plant, are also haploid, and each archegonium has an egg. And as we kind of talked about a little bit before when we were talking about seed plants in general, is that this is really a cool evolutionary leap because look at where the eggs are located, right? The eggs are located in the female gametophyte plant that is now inside of this ovule structure. So it's this whole layer of protection. Look, female gametophyte plant in the middle. First of all, it's surrounded by the nucellus or the Microsporangium. So it's surrounded by that. And then on top of that, it's surrounded by many layers of this integument. So that's a lot of protection for these eggs inside. from where we saw before, way back in the non-vascular plants where eggs were just on a free living gametophyte plant living out in the world. Now, gametophyte plant is inside an ovule and protected by all of these layers. So that's huge for plant development. And this is part of the reason why we have so many ovule / seed bearing plants, because this system works really well. If you protect the egg better, you have more success over time, more fitness, better protected egg, better protected zygote, more offspring over time. So, so here's your ovule, right? With the parts waiting. So, right, here's your ovule with the female gametophytes and female gametophyte inside with her archegonia and egg, right? Here it is. And what's going to happen is on another cycad plant, the male cycad plant, the Microsporangia, aka pollen sacs, are going to open and release pollen grains out into the wind. And the pollen grain, if it's lucky, it will land on an ovule. Let's do this. Here's pollen grain. See, okay, so it says pollen grains. Oops, that ain't great. But each of these little dots, I don't know if I could do this. 1, 2, oops, that didn't work. So let's try this again. Pollen grains. Each of these little dots, 1, 2, 3, each of those little dots is a pollen grain, and which would have come from a pollen sac. And if the pollen grains are lucky, they'll land on an ovule. Oops, part of the way that they part of the way their luck increases is that the ovules secrete this thing, which is called a pollination drop. So at the entrance of the micropyle, this little wet drop will, when the ovule is ready, will come out, and then that will help capture pollen grains that are floating by in the wind. So yeah, what does it say here? The ovule produces a sticky drop, right? The pollination drop that emerges from the micropyle. Pollen grains from the Microsporangia, aka pollen sacs, if they're lucky, will land on the pollination drop. And this process is pollination. So if I ask you ever on an exam about the process of pollination, pollination is literally when a pollen grain lands on an ovule. Done. Pollination complete. So pollen grain lands on an ovule, that's pollination. That's different from fertilization. Fertilization is fusion of egg and sperm. Pollination is pollen grain landed on an ovule. Okay, what's next? All right, so let's go through what happens. And my heat is turning on, so if you hear some loud noises, I apologize ahead of time. Once that pollen grain lands on that pollination drop, which is coming out of the ovule, eventually that pollination drop will dry back up into the ovule. And when that happens, it'll take some pollen grains in with it. So here is the entrance. So the pollination drop would have occurred right here at the entrance of the micropyle. And as I just said, the pollination drop will dry up and bring the pollen grains into the ovule through this micropyle, through this hole. When the pollen grain, via that pollination drop that's drying up and going back into the ovule, when it comes down the micropyle, the first place that the pollen grain enters is this place called the pollen chamber. And Same process is again what happens for all the gymnosperms. So there'll be a decent amount of repetition on this process as we go forward in the different lectures. But yep, so pollen grain was stuck into the pollination drop. Pollination drop dried up and brought the pollen grain down through the micropyle, which allows the pollen grain to arrive in the pollen chamber. Once that pollen grain is inside of the pollen chamber, it's going to grow that pollen tube cell that we talked about before. And that pollen tube cell is going to grow through the wall of the Megasporangium, aka nucellus. It's going to grow through that. Once the pollen tube cell has grown all the way through that Megasporangium wall, then the two sperm can swim down the pollen tube cell. So here we see pollen tube cell. It's a funky drawing. I didn't draw this. But the point of this drawing is this green colored stuff is the wall of the Megasporangium that's inside of the ovule. This gray stuff is the pollen tube cell that has grown. And these little squigglies are the sperm. So here, the pollen tube cell was in the pollen chamber out here. It grew pollen tube cell to enter the, Megasporangium, went through. Inside that Megasporangium is the female gametophyte plant. There's a question here. Is it usually only two sperm? Each pollen grain produces two sperm. And each pollen grain produces one pollen tube. So yes, for each pollen grain, there will be two sperm per pollen grain, which we'll talk about as we as we learn more about the evolution of seed plants. This system of two sperms is kind of wasteful because even though there are two eggs in the female gametophyte plant, ultimately only one zygote will win. Meaning that, so here's your pollen tube cell, and your pollen tube cell contains two sperm. But of these two sperm, this pollen tube cell is not going to fertilize both of these eggs. This pollen tube cell is going to release the sperm, and the sperm are going to compete for the same egg. And whichever sperm is faster and better and stronger is going to be the one that ultimately fertilizes the egg. It might be this egg, it might be this egg. Don't worry about that right now. I'm just saying it's a little wasteful to have two sperm because ultimately only one gets used. So swimming sperm are now going to enter the archegonial chamber. So this diagram here kind of brings us back to where we were before. Here's that hole, the micropyle, which is what had a pollination drop, which drew the pollen grains into this pollen chamber. Once in the pollen chamber, pollen grains can grow a pollen tube to grow through this wall of the through this wall of the Megasporangium, which is this hatched thing here. And once the pollen tube cell grows through that wall, it will allow the sperm from the pollen grain to be released into what's called the archegonial chamber, where the archegonia with egg are waiting. So this is just showing you where that archegonial chamber is located with respect to the other places we've talked about so far. Okay, we're almost done. Okay, so remember that pollination is when a pollen grain lands on an ovule, whereas fertilization is when one sperm fuses with one egg. And as I just mentioned, you have originally in one pollen grain 2 sperm, but only one sperm will fertilize one egg. The other sperm will degrade, and the other egg will go unfertilized. And this is part of the reason why cycads are endangered, as we talked about at the beginning of this lecture, is because it can take months between pollination and fertilization in the cycads. So what does that mean? It means that a pollen grain could land on an ovule and fertilization won't occur for months after that. It has a really, really slow reproductive rate, which is one of the reasons that it's facing extinction in some areas. And we talked about that at the beginning of this lecture of reasons why cycads are facing that. And this is one of the reasons. It's very, very slow reproduction. Do you guys remember way back in lectures where I would always say you have mitosis and cytokinesis, or meiosis and cytokinesis, and cytokinesis happens usually but not always right following mitosis. Now we're going to actually see an example of that. So when the egg and sperm come together in a cycad ovule, the result is a 2N zygote, just like we've seen before. However, what's going to happen now is so egg and sperm come together and results in a 2N zygote. But that 2N zygote is actually going to undergo lots and lots and lots and lots and lots of mitosis without any cytokinesis initially. And this is something we call the free nuclear stage. Here is an example of what that looks like. So here on the left, you have one fertilized egg. So this whole circle, or this whole ovule, this whole oval is an egg. And then we know it's been fertilized because we can see the nucleus in the middle, which is the result of egg and sperm. So we can think of this as the beginning of a zygote, this whole thing here. There's going to be lots and lots of mitosis occur. So a lot of cell division is going to occur, but no cytosis initially is going to occur. So you see this going from one nucleus to many nucleus, all still within one cell. Later, because of different chemical signaling, the nuclei are going to become arranged. Once they become arranged in the proper places, then finally we will see cytokinesis occur. So this is why we always, when we talk about these processes, we always say it's mitosis with or without cytokinesis, because we do have cases where mitosis occurs and we don't have any cytokinesis. This same process of this like free nuclear stage also occurs in animals, just so you know. So like if you study fruit flies or something like that, you may have already known this because this is something that happens in them. Okay, almost done. Right. So this is what I just said. You have that free nuclear stage, and then the nuclei get organized. Cytokinesis finally occurs, and then the final result is an embryo, and the embryo is going to continue to grow via mitosis inside of what is now the seed. It was an ovule with an egg. Yes, so Ashley's asking if we have 20 minutes for the quiz, right? Yes, don't worry, we're going to start the quiz very soon, and it shouldn't even take you 20 minutes, but it'll be open for all of the rest of the class time. We're almost done here. Yeah, so the embryo is going to continue to grow, which is inside of what is now the seed, right? It was the ovule with an egg. Now it's an embryo with a seed. No, it was an ovule with an egg. Now it's a seed with an embryo. We don't have to spend much time on this today, but just know that there's different parts of an embryo, including embryonic leaves, what will become the roots and the shoots, parts of the stem, and embryonic root. We'll talk more about that another time. But the main take-home message here is, so the green thing here is the embryo, and the blue stuff around it is the female gametophyte plant. And in gymnosperms, the embryo consumes the female gametophyte plant as a food source as it grows. Yep. Eventually, once the embryo is large, so here's an example. This is what the seeds look like. Each of these seeds have embryos inside. Those seeds are going to mature on the sporophylls of either strobilus or fertile crown. And when the embryo gets large enough, the seeds will fall off and the embryo will come out of the seed will germinate and grow into the new sporophyte plant. So that is the cycads. We've covered them now. We're finished with them now. And the next class, we're going to go through the next part of the gymnosperms, which are ginkgo biloba. So if you guys have any questions right now, I'm happy to answer them. If not, if you don't have questions now, or if you do have questions now and you just don't want to answer them or ask them because you're like thinking about the assignment, that's fine. Send me any follow-up questions that you might have via email. I should also mention I still have technically office hours, so if you ever want to meet one-on-one to discuss anything about the course, we can meet here in this room, this virtual room, for office hours. So keep that in mind, okay? You're not just on your own now because we're virtual. I still have office hours, so happy to meet with you if that would be useful to you. Okay, so what I would suggest now is that everybody should go on to Blackboard. And I'll just open the assignment a little earlier. I think I can do that. Oops. Oh, geez. 1 second. Mm-hmm. One second. I think I can. Yeah, sorry. It makes you do it in all these stages. Oh, geez. Oh, maybe this? Nope, that one. Here, let's try that. Ah, here we go. There we go. All right, so I'm going to just make it available. Okay, submit. Cool. So if you log on to Blackboard right now and go to assignments, you should see assignment 4 there. Okay? So good luck on the assignment. I'm going to send you guys the homework assignment later, which is to do that little practice thing to understand that platform. And yep, good luck with the assignment. And let me know if you have any questions. And I'll talk to you guys later, and I'll see you next class.